Messenger RNA, or mRNA, is a crucial molecule in our cells that carries genetic instructions from DNA to the protein-making machinery. To understand mRNA, let's first look at a cell. The DNA in the nucleus contains our genetic information. Unlike DNA, which stays in the nucleus, mRNA is created as a temporary copy of a specific gene through a process called transcription. The mRNA then travels from the nucleus to the cytoplasm, where ribosomes read its sequence to build specific proteins. This entire process allows our cells to use the genetic information stored in DNA to build the proteins needed for nearly every function in our body. Let's compare traditional vaccines with newer mRNA vaccines. Traditional vaccines typically contain weakened or inactivated viruses or fragments of viral proteins. In contrast, mRNA vaccines do not contain any part of the virus itself. Instead, they contain messenger RNA, genetic instructions packaged in lipid nanoparticles. With traditional vaccines, the immune system directly recognizes these viral components as foreign and mounts an immune response. In contrast, mRNA vaccines work differently. Once inside our cells, the mRNA provides instructions for the cell to temporarily produce a specific viral protein. The key difference is that mRNA vaccines never introduce the actual virus into the body. Instead, they use our own cellular machinery to produce just one specific viral protein, which then triggers the immune response. This innovative approach allows for rapid vaccine development while maintaining safety, as no actual viral particles are ever present in the vaccine. Scientists begin by analyzing the virus structure to identify the most suitable protein to target with the vaccine. For coronaviruses, the spike protein is the ideal target because it's essential for viral entry into cells and is easily recognized by the immune system. Once the target protein is identified, scientists use computers to design an mRNA sequence that will code for that protein. The mRNA sequence contains the exact instructions for human cells to produce the viral spike protein, using the genetic code of A, U, G, and C nucleotides. After designing the mRNA sequence, scientists synthesize it in the laboratory using specialized enzymes. This process builds the mRNA strand nucleotide by nucleotide following the exact sequence designed on the computer. The final step involves modifying the mRNA to improve its stability and optimize protein production. Scientists add protective elements, like a cap at the beginning of the mRNA and a poly-A tail at the end. They also replace standard nucleosides with modified ones to reduce unwanted immune responses and improve translation efficiency. mRNA on its own is extremely vulnerable in the body. Without protection, it would be quickly degraded by enzymes. These enzymes break down the mRNA strand into smaller fragments, rendering it useless before it can reach cells and deliver its instructions. To solve this problem, scientists encapsulate the mRNA inside lipid nanoparticles, essentially tiny bubbles made of fat molecules. These lipid nanoparticles form a protective barrier that shields the mRNA from degrading enzymes in the bloodstream. These nanoparticles are crafted from different types of specialized lipids, each with a specific function. Ionizable lipids help the mRNA escape from cellular compartments, phospholipids form the structural bilayer, cholesterol provides stability, and PEG lipids prevent the particles from clumping together. When the lipid nanoparticle reaches a cell, it can fuse with the cell membrane. This is possible because both the nanoparticle and cell membrane are made of similar lipid materials. The nanoparticle approaches the cell membrane and begins to fuse with it. The lipids in both structures rearrange to allow this fusion process. During fusion, the nanoparticle's lipid bilayer merges with the cell membrane, creating an opening that allows the mRNA to be released inside the cell. In summary, lipid nanoparticles are essential for mRNA vaccine delivery. They protect the fragile mRNA from degradation and help it enter our cells safely, ensuring the genetic instructions reach their destination intact. Once the vaccine mRNA enters the cell, it encounters ribosomes in the cytoplasm. Ribosomes are cellular machines that read the mRNA instructions to build proteins. The ribosome attaches to the mRNA strand at the start codon and begins reading the nucleotide sequence, 3 letters at a time. As the ribosome moves along the mRNA, it recruits amino acids that match each codon. These amino acids are joined together to form a growing protein chain. Once the complete amino acid chain is formed, it begins to fold into its functional three-dimensional structure. For the COVID-19 vaccine, this results in the characteristic spike protein. The newly created spike protein is then transported to the cell surface. There, it will be displayed on the cell membrane where immune cells can recognize it. After cells produce viral proteins from the mRNA vaccine, these proteins are released into the extracellular space. Dendritic cells, which are specialized immune cells, patrol the body searching for foreign substances. When dendritic cells encounter these viral proteins, they capture and engulf them through a process called phagocytosis. Inside the dendritic cell, the viral proteins are broken down into smaller fragments called antigens. These fragments are then loaded onto special molecules called MHC class II, which transport them to the cell surface. T cells circulate in the body and can recognize these presented antigens. When a T cell's receptor matches an antigen presented by the dendritic cell, recognition occurs. This recognition activates the T cell, causing it to multiply and triggering a cascade of immune responses. Meanwhile, B cells also play a crucial role in the immune response. B cells can directly recognize viral proteins. When activated T cells interact with these B cells, they enhance the B cell response. This interaction between dendritic cells, T cells, and B cells establishes a coordinated immune response against the viral proteins produced from the mRNA vaccine. After the body mounts an immune response to a pathogen, most activated B and T cells will die off. However, some B and T cells transform into specialized memory cells that can survive in the body for decades. These memory cells take up residence in lymph nodes and circulate through the bloodstream, constantly patrolling the body for signs of the same pathogen. When the same pathogen enters the body again, these memory cells recognize it immediately and mount a much faster and stronger immune response. This graph shows the difference between primary and secondary immune responses. During the first infection, the antibody response takes about a week to develop. But during a second encounter with the same pathogen, memory cells activate immediately, creating a faster, stronger, and more effective immune response. This memory cell system is what provides long-term immunity after vaccination or natural infection, allowing your body to quickly defend against pathogens it has previously encountered.